So I'm very pleased to introduce our today's seminar speaker, Dr. Oscar Monroy. Dr. Monroy has spent much of his scientific career exploring aquaculture nutrition and aquatic ecosystems, specifically helping to develop novel molecular technologies to aid in the study of lipid and fatty acid biochemistry. Dr. Monroy earned his doctorate degree from the University of Valencia in Spain. He then worked as a postdoc all over Europe, including the Instituto Español de Oceanografía, Bangor University, University of Stirling, and the Institute of Aquaculture Torre de la Salle. The last of which now serves as his home, where he is a tenured scientist. And his research interests include the study of molecular mechanisms involved in the biosynthesis of long-chain polysaturated fatty acids and aquatic organisms and their implications in nutrition of farm species. So thank you so much, Dr. Monroy, for agreeing to speak for us today. I'll turn off my camera and mic and pass it off to you to share your screen. Thank you.
Okay. Thanks, Ruba. Thanks very much for the introduction. So I'll be start sharing my screen. So can you see it?
Yeah, we can see
it. Thank you so much. Okay, thanks very much, Ruba, for the introduction, and also thanks to everybody for having me today. It's an honor to be presenting here at this seminar series. So as Ruba said, my name is Oscar Monroy, and I'm a tenured scientist at the Institute of Aquaculture Torre de la Salle. And today I'm going to be talking about biosynthesis of long-chain polyunsaturated fatty acids in aquatic invertebrates. I'll be showing you what we know about the genes involved and which functions their encoding enzymes have. Okay, so just a brief introduction of where we are. I'm actually speaking to you from home today. But I work at the Institute of Aquaculture Torre de la Salle, which belongs to SCIC, which is the Spanish National Research Council, which is the largest research institution in Spain, and it's got like 120 institutes. And Torre de la Salle is the only one devoted exclusively to marine aquaculture research. So we are sitting on the east coast of Spain, in Valencia region, and this is an old picture of Torre de la Salle. Now it's more populated with buildings. It's a pretty touristic place, so it doesn't look anymore like that, but still a nice location. Okay, beside the sea, Mediterranean Sea. Okay, so this is the outline of the talk today. I'll be starting with a bit of background, some definitions of long-chain PUFA, what these compounds do or where they come from. And then I'll move on what we know or how how research in this field started back maybe 15 years ago on fish, characterization of the saturases and elongations involved in long-chain PUFA biosynthetic pathways in fish, in farm species in particular, and how we move from fish research to invertebrates, a topic that started like 10 years ago, more or less, okay? So invertebrates enzymes will be the core of this talk, and we'll be covering three main types, starting with front-end desaturases, FAD, okay, which are desaturases that we also have in fish. And so we will move to elongases, the same thing, the same case with these genes also occurring in invertebrates like fish. And then we'll move to a third type of enzymes, what we call omega-X desaturases, which are enzymes that are absent from vertebrates. So fish do not have it, mammals do not have it either. So these are a bit of a particular special case within these pathways that we're going to be seeing today. Okay, so what I'll be referring to as long-chain polyunsaturated fatty acids. So polyunsaturated fatty acids are fatty acids that have at least two unsaturations, okay? And unsaturations mean double bonds within the acyl chain of the fatty acid, okay? For instance, the top fatty acid is alpha-linolenic acid, which is a PUFA. It's a polyunsaturated fatty acid with 18 carbons. And it belongs to the N-3 series. That means that it's an omega-3 fatty acid, which will be the main characters here in today's talk. Okay, but it's not a long-chain PUFA according to the definition I have just given to you. So long-chain PUFA are PUFA with 20 carbons or longer. Okay, so the top structure, the top fatty acid, alpha-linolenic, is not a long-chain PUFA, whereas the bottom, the docosahexaenoic acid, 22,6 omega-3, is a long-chain PUFA because it's got 20 or more carbons. In particular, DHA has 22 carbons. So bear in mind that there is both of these examples are omega-3 fatty acids, but only the bottom is a long chain PUFA, whereas the top one is what we call a PUFA, a simple PUFA. Okay, so the DHA and other long chain PUFA have biological functions, important biological functions, whereas the PUFA are just mere precursors of the long chain, as I will explain later. Okay, so they don't play significant or important biological functions in comparison to DHA and other long chain PUFA. Okay, so what are omega-3 long chain PUFA? So they are natural lipids that are mostly derived from the marine trophic webs. Okay, so they are produced at the bottom of the seafood web by mostly single cell organisms, but not exclusively. And this is why the asterisk stands on the second bullet point. And I will get back to this. We have discovered that along single cell organisms, like microalgae, protists, etc., many different invertebrates can also contribute to omega-3 long chain PUFA production in the ocean. Okay, so I will get back to this later on in my talk. So omega-3 produced at the bottom of the seafood web gets accumulated as we go up to the food web, and fish, for instance, and seafood in general become primary sources of omega-3 for human consumers. Okay, so fish, particularly oily species like, for instance, sardines, anchovies, mackerels, etc., accumulate this because every single food item in the food web contains omega-3, and these compounds get accumulated as we go up in the web. Okay, so as I said, DHA is one of the most important omega-3 long chain PUFA, but there is another one which deserves attention, which is EPA. EPA is a 20 carbon long chain PUFA, and together with DHA play important roles not only in invertebrates in general, they have been also seen beneficial with in some human pathologies like the ones that are listed here. So omega-3 have been seen beneficial in cardiovascular disease and also other inflammatory diseases. DHA in particular plays important roles as in nervous system functioning, simply because it's a main component of nervous cell membranes, and therefore ensures that the nervous system functions correctly. And omega-3 long chain PUFA in general have been seen also beneficial in some types of cancer, like prostate cancer, depression, Alzheimer, etc. So overall, they are important components in our diet, and we used to get them by consuming farm fish. Okay, simply because the diets that we used to use in fish farming contain omega-3 long chain PUFA, and those omega-3 long chain PUFA derive or were available in the diet because in those diets that we used to use in the past were composed mainly by two compounds, two ingredients, fish meal and fish oil. From now onwards, I will be exclusively focusing on the oil component of the diet, fish oil. And fish oil, deriving from, for instance, captured fisheries like anchovy, the most important fisheries worldwide, provides omega-3 long chain puffer because of the reasons I explained to you earlier, simply because they accumulate them through the diet, and they accumulate this in the oil component in the oil, and then when we extract the oil from the anchovies and other feed grade fisheries, we have this oil being rich in omega-3 long chain puffer. Okay, so when we formulate diets for fish farming with fish oil, those omega-3 long chain puffer that are present in the diet accumulate in the fish. So those fish produced that way are omega-3 rich, and therefore they provide the benefits that I was referring to in my previous slide. Okay. Not only that, we ensure the quality of the food, of the fruit product for human consumers, but we also guarantee the nutritional requirements in terms of essential nutrients being covered. Okay, so we cover both sides very, very nicely, with including fish oil apart from fish meal. But as you well know, there is a limited production of fish oil, and that applies also to fish meal. And this production annually, globally speaking, is roughly 1 million tons, not even that much, okay? And that was enough some years ago, but with expansion of aquaculture, particularly with carnivorous species, that amount that remains limited, that fixed, let's say, has to be distributed for producing much more fish, okay? So other strategies have been necessary to be put in place. And nowadays, the fish oil that we used to put in the diets has been reduced and replaced with other sources. And these other sources replacing fish oil have been primarily vegetable oils. Problem with using vegetable oils is that they are devoid of long chain puffer. Naturally, they don't contain long chain puffer. And this has two main effects. On one hand, we produce fish that accumulate less amount of omega-3 long chain puffer, because simply the diets do not contain so much, and we are diluting, or we are reducing the nutritional quality of those fish simply because they don't contain so much omega-3 long chain puffer. Okay, this one is one of the effects. But the second effect is on the fish health itself. Okay, because if we are replacing too much fish oil and adding too much vegetable oil in species that do not handle this dietary challenge very well, we are potentially compromising their health and their growth. Okay, so this is why we started investigating how fish could cope with this challenge, with this dietary challenge of changing so dramatically the fatty acid composition of the diets. And lipid metabolism is a main actor, let's say, in this story. Within the different lipid pathways, lipid metabolism pathways that explain how fish can utilize efficiently C18 puffer containing vegetable oils to provide themselves with the biologically essential long chain puffer, there is one pathway that is particularly important, and this is the long chain puffer biosynthesis, which is going to be the pathway I'm going to be focusing on from now onwards. Okay, so a little bit of biochemistry here. So we have this pathway. So the biosynthetic pathways of long chain puffer can be described as consecutive reactions catalyzed by two enzymes. On one hand, we have desaturases, which introduce new double bonds, new unsaturations, and then we've got elongases, which expand the length of the pre-existing fatty acids in 2 carbon units. Okay, so the action of these two enzymes, desaturases and elongases, enable the conversions of from C18 puffer. So these are the 2 precursors of the pathways. So we've got alpha-linolenic acid as precursor of the N-3 or omega-3 pathways at the bottom of this slide. Okay, so this pathway here. and then linoleic acid 18,2 omega-6 or 18,2 omega and N-6 in the N-6 series, in the omega-6 series, okay, at the bottom of this slide. So these two C18 PUFA are the precursors, and through the action of desaturases and elongases, there are a series of conversions leading to the production of EPA and DHA in the omega-6 series above and arachidonic acid at the bottom pathway, so the N-6 pathway, okay? So the ability of a fish to go from C18 PUFA precursors to long-chain PUFA, including EPA and DHA in the N-3 series and arachidonic in the N-6 series, explains how capable those fish are of utilizing vegetable oils rich in the C18 PUFA and to satisfy requirements of the biologically important EPA, DHA, and arachidonic acids. So these pathways have been investigated in many, many species. Okay, there are some examples here. And you will probably have read at some point that freshwater species and salmonids have high capacity to do those conversions I was showing you in my previous slide, whereas marine species have lost such capacity simply because they satisfy their requirements through food items that they consume in the sea, and they have lost, and therefore they have lost such capacity, the capacity to produce the C18, sorry, the C20 and C22 long-chain PUFA from the C18 precursors. This is no longer, such dichotomy is no longer accepted, or it's regarded as a bit simplistic, and it's more complex than that. But the idea is that for any particular species, we need to know the complement and function of desaturases and elongases to fully understand how capable they are to do the conversions I showed you earlier. So just to give some details, desaturases, there are several types of desaturases. And in this example, we see that there are two types. We call them delta-6 desaturases, operating at the beginning and at the end of the pathways, and a different front-end desaturase operating these pathways is the delta-5 desaturase, which catalyzes reactions sitting in the middle of the pathways. There are other front-end desaturases, and fish do have other front-end desaturases. I didn't put them on the diagrams just to simplify the story a little bit, but there are also, apart from delta-5 and delta-6 desaturases, we've demonstrated the existence of delta-4 and delta-8 front-end desaturases. But anyway, the thing is that they belong to a single family, which we call generically fats, okay? Apart from fats, from desaturases, we've got elongases, and these are the reactions that catalyze these enzymes, okay? So the action, the coordinated action of desaturases on one hand and the elongases enable a species to go from C18 PUFA to the C20 and C22 long-chain PUFA. With regards to elongases, there are several types. We've got EloVL5, which operate at the early stages of the pathway. So this is only the omega-3 pathway. So I have removed the omega-6 pathways for simplicity. But this LOBL5 and other elongases operate exactly the same point in the other pathway, omega-6 pathway. So LOBL5 elongates C18s and C20s. LOVL2, which is another PUFA elongase, elongates primarily C20 and importantly C22. This is a very important step, elongation step to produce DHA, which is this compound here, okay? And then we've got LOVL4, which in addition to producing what we call very long-chain polyunsaturated fatty acids, so fatty acids that can reach up to 38 carbons, LOVL4s can also help LOVL2 in the production of 24 PUFAs from C22 substrates, as I explained for LOVL2, okay? So these are the three main PUFA elongases that operate in fish. Okay, so let's move now to in invertebrates. So this research, so the transition from fish to invertebrates started like 10 years ago, roughly, when we were involved in a project trying to figure out the essential fatty acids that cephalopod paralarvae have. Okay, so I don't know whether you are familiar. Well, Octopus vulgaris, the common octopus, is a species that is a candidate species to diversify Mediterranean aquaculture. So in Spain, Portugal, Italy, we eat octopus, and it's a very traditional food item, okay, seafood item. Paralarvae die just by looking at them. They are very, very tricky to maintain. And obviously that complicates running feeding trials in which we can test the requirement of those paralarvae. Instead of running feeding trials, which, as I'm telling you, they are very difficult to carry out, we had a different approach, an approach by which we isolated the genes, we constructed those pathways, but in this species, in Octopus vulgaris, and to see where were the gaps, okay, the gaps in which we needed, so the octopus wouldn't have any gene, and instead we needed to provide the corresponding fatty acid preformed in the diet to ensure there is no deficiency symptoms. And this is our results, okay, or a summary of them at least. Initially, we cloned a gene, an elongase, which we called LOVL25, and this name derives from the fact, well, it's explained by the fact that in fish, for instance, as I told you earlier, we've got two separate proteins, LOVL2 and LOVL5. In invertebrates, these two proteins exist as one single protein, and that's why we call it LOVL25. Functionally speaking, LOVL25 behaves more or less like an LOVL5, a vertebrate LOVL5, a fish LOVL5, very actively or very efficient in elongation of C18 and C20 PUFA, okay? So catalyzing the initial elongation steps in the pathways. Then we characterized an LOVL4, okay, which in addition to producing very long chain PUFA, and I'm not showing this on this diagram, they also, or LOVL4 from octopus, catalyze the elongation of C22 to C24. So again, potentially contributing to the biosynthesis of DHA, okay? We characterized a front-end desaturase that happened to be a delta-5 desaturase, and therefore enabling this reaction and this other reaction. But that was it, okay? So these were the only three genes that we could find at that point. And as you can see, we have interconnected reactions going from this fatty acid 18,4 omega, so 18,4 omega-3, sorry, okay, and all the way to 24,5 omega-3. However, there are gaps here, reactions that were not detected, or we couldn't find any enzyme explaining those reactions. And therefore, this type of information enabled us to, for instance, identify this compound, DHA, as a dietary essential fatty acid that must be provided in the diet of Octopus vulgaris paralarvae to ensure paralarvae do not show deficiency symptoms, okay? So this is the sort of information we can gather by looking into the genes and by characterizing the functions of the encoded enzymes. Okay. So moving into the first set of enzymes that we will be seeing in invertebrates, front-end desaturases. Let me explain first how they operate. So front-end desaturases are enzymes that are present in fish, invertebrates, okay? And they are also present in invertebrates as well, okay? The delta-5 desaturase of octopus, for instance, is a front-end desaturase. The delta-6, delta-5 desaturases operating in fish are also front-end desaturases. And the way these enzymes operate are as follows. We've got a substrate, like for instance, linoleic acid, 18-N-6, okay? And a delta-6 desaturase introduces a new double bond, a new unsaturation at the carbon number 6, that 6 being the 6th carbon counting from the carbon of the carboxyl group at the front end of the fatty acid. Okay, so we count 1, 2, 3, 4, 5, 6, and then a new double bond is introduced by a delta-6 desaturase. Delta-5 desaturases, delta-4s, and delta-8s operate in a similar manner, but introducing the new double bond in position 4, 8, or 5 as corresponds, okay? Okay, so what about the front-end desaturases that we have found in invertebrates? Let me show you what we have characterized by showing the different types of front-end desaturases from using these three as an example, as a model of showing how many the different subtypes of front-end desaturases we have found in invertebrates, different subfamilies of front-end desaturases. Okay, on one hand, we have this type A, okay, which encompasses only exclusively delta-5 desaturases. And we have found delta-5 desaturases in different mollusks, including the Octopus vulgaris I mentioned earlier, and also other mollusk species. And then we have found delta-5 desaturases in Paracentotus lividus, okay, echinoderm, which has 3 desaturases, and one of them is a ape-type front-end desaturase, all also being delta-5 as the mollusk ones, okay? So in this group, we have only delta-5 desaturases. In type B, okay, we have a variety of subspecificities. So we've got delta-8, and we can have also delta-6 desaturases. There have been only 2 desaturases being characterized from this group, both from mollusk species, okay? And then we have a different type which includes exclusively echinoderm sequences, and the 2 that we have characterized have been only delta-8 desaturases. We don't know whether there are other functionalities, but the only 2 we've characterized, they are delta-8 desaturases. So the idea here is that we have 3 different families. Remember that in fish, in teleost, we have only 1 type. In invertebrates, we have 3 different subfamilies. At least, phylogenetically speaking, they are related, but different altogether. So apart from echinoderms, mollusks, etc., there are front-end desaturases in sponges. So at the very ancestor of the animals, we have already sponges, but they haven't been characterized functionally. The function hasn't been characterized, as I explained earlier, for other invertebrates. However, there is a good body of knowledge and evidence, molecular evidence, that sponges have front-end desaturases, and this is supported by the presence of puffa of fatty acids in the sponges, fatty acids that we know as demospongic acids, which are characterized by having delta-5 and delta-9 desaturation and saturation patterns. So the presence of this delta-5 unsaturation in puffa from sponges indicates the presence of delta-5 desaturases, okay? But they haven't been characterized yet. in sponges. And this is pretty much the case in other invertebrates like Nigerians. Okay, so this paper reports on the presence of fatty front-end desaturases like sequences from Nigerians, but they haven't been characterized yet functionally. What about crustaceans? These are 4 different papers claiming to the presence of delta-6-like desaturases from different species of crustaceans. However, any of these papers actually provided functional evidence. So we don't know whether these desaturases claim to be delta-6 desaturases are actually delta-6 desaturases, okay? However, some studies, for instance, in this species, in this paper, this crayfish species presented gene expression data that suggests that these delta-6-like desaturases behave similarly to what we would be expecting from a front-end desaturase. And this behavior is illustrated here in these 2 figures. On the left-hand side, we have crayfish being fed with different diets. So we can see like the vegetable oil-rich diets, which is the SO diet, produced an increase in the expression of this delta-6-like desaturase. And that means that the crayfish responds with increasing expression to compensate, to produce more long-chain pupa because the diet contains low levels, okay? And this is how front-end desaturases behave. And also, when crayfish are cultured at low temperature, there is also an increase in the expression, and this is common in invertebrates as a mechanism to make membranes more fluid by unsaturating fatty acids in the membranes and making the membrane more fluid and accommodating membrane-bound proteins and other molecules. Okay, so the way this delta-6-like, despite not being functionally characterized, behaves very similarly to front-end desaturases. So probably these crustaceans also have delta-6 desaturases. Okay, so moving into elongases, remember that elongases are also present in vertebrates. So front-end desaturases and elongases exist in fish, in other vertebrates as well, like mammals, etc. And this tree is helpful for illustrating which types of elongases, pupa elongases, we have found in invertebrates. For instance, we have found LOBL25, as I told you earlier for the case of octopus LOBL25. LOBL25s are very efficient in elongation of C18 and C20s, okay? And we have found it in different mollusks and also in the invertebrate chordate Amphioxus. Then another type of pupa elongase is the LOBL4, which is efficient in the elongation of pufas with chain length of C18, C20, and C22, and it's been characterized in mollusks and also tunicates. And then, apart from LOBL25 and apart from LOBL4s, we have detected, or it's been detected, not ourselves, this is a work from a group in Canada, detection of putative LOBL4s in sponges, again, Nigerians, and also mollusks, okay? And I like to bring up the attention on 2 different subfamilies of what they refer to as novel elongases, okay? Novel LOBL-A and novel LOBL-B. So Nigerians, and this is the case also for other invertebrates, in addition to having LOBL25, LOBL4, have also different subfamilies of elongases. And potentially these novel elongases, which haven't been characterized yet, contribute also to the PUFA production. So contribute to long-chain PUFA biosynthesis. Okay, and this is again illustrating how diverse the families of enzymes, in this case elongases, is in invertebrates in comparison to what we find in fish, for instance. Okay, again, crustaceans, there have been gene expression experiments suggesting that, again, crustaceans have PUFA elongases, judging from how these elongases respond diet composition of low long-chain PUFA inclusion and upregulation of the gene, trying as a mechanism to compensate lack of dietary provision and endogenous production. And this is very similar to other PUFA elongases from both fish and invertebrates. So it's likely that many of these, despite not being characterized in crabs, these PUFA elongases are actually active on and contributed to the long-chain PUFA biosynthesis. Lately, we have characterized, this is a paper published earlier in 2021, in which we have reported the presence of 3 different elongases in marine gammarids, and the capacity of, or subsidy specificity of these 3 elongases enable marine gammarids to perform all the elongation reactions in the pathways I showed you earlier. However, we are not certain about the capacity of desaturation, and this is work in progress. We don't know. So gammarids can do elongation pretty well, but we are not so sure about how capable of desaturation they are. Okay. So as I showed you earlier, we've seen 2 gene families, front-end desaturases and elongases, that exist in invertebrates, but also exist in vertebrates. In fish, in invertebrates like fish, mammals, etc. But this 3rd family, omega X desaturase, doesn't exist in invertebrates. And actually, it was believed for a long time that it didn't exist in animals, in invertebrates either. Okay, so that has like a big discovery that we had the lab to contribute to. Okay, so the way omega X desaturases operate is as follows. So in contrast to front-end desaturases that introduce the double bond in this site, in the front end of the fatty acyl chain, methylene desaturases introduce the new double bond in the other side, counting from the carbon of the methyl end at one of the ends of the fatty acid. So this makes that the action of an omega desaturase, also called methylene desaturase, makes that a fatty acid which is omega-6 becomes an omega-3 product. Okay, so for instance, the action of an omega-3 desaturase converts linoleic acid 18-2 omega-6 into an omega-3 product, alpha-linolenic acid 18-3 omega-3. So that makes that, well, these desaturases, omega-X desaturases, were discovered in a paper we published 3 years ago in many different species of invertebrates, including nematodes, corals, rotifers, annelids, crustaceans, etc. So a total of 80 species and a total of 121 sequences were identified, forming different clades. We don't know the relationship between the 3 types, but functionally they are very similar, and they all have been demonstrated to be omega-X desaturases, many of them being omega-3, as the example I showed you later. So what happens with an animal that has omega-3 desaturases or omega-X desaturases in general? So this slide shows the biosynthetic pathways of long-chain PUFA in a different way as I showed you in previous slides. So we've got the N-6 series pathway here. Then we've got the N-3 pathway here. And then we've got a pathway of de novo synthesis. So animals in general can produce saturated fatty acids with a complex enzymatic complex called fatty acid synthase. And then animals and organisms in general have desaturases that we generally refer to as first desaturases. So the desaturases that introduce the first unsaturation, so producing monounsaturated fatty acids. So the idea is that we have three different pathways which are not connected to each other. Okay, so we've got this pathway on one hand, we've got this other pathway, and separated this third pathway. Okay, so when an animal has omega X desaturases, these pathways get connected. Okay, so how animals can do that? So these are separate items, separate, we call it metabolic islands, let's say. So the existence of omega X desaturases, like for instance, an omega-6 desaturase, connects one pathway with the omega, so the de novo synthesis pathway together with the N-6 or omega-6 pathway. And then the existence of omega-3 desaturases connects these two metabolic islands that were joined together by the action of the omega-6 desaturase with the N-3 series. So everything gets connected here. So that in theory enables an animal to go from short chain saturated fatty acids, okay, of very short chain, all the way to long chain PUFA, okay? So having this capacity is very attractive, very interesting, and we exploit this capacity in two different ways. Okay, so why having these desaturases is so important? Why this is such big deal? Okay, so in one hand, the dogma by which only single cell organisms contribute to the production of omega-3 in the marine ecosystem is no longer valid, because many different invertebrates can also potentially contribute to the production of omega-3 in the ocean. Okay, so the discovery has an impact at the ecological level, but also something more applied that we are now exploiting in different projects, as I will explain you next. The capacity of an animal to have omega-3 desaturases enables the culture of this animal to produce omega-3 rich biomasses that can be used as an ingredient to formulate fish. So polychaetes become new sources of omega-3, and we believe that this is a good strategy to compensate that limitation of omega-3 rich ingredients that are nowadays limiting the production of feeds for aquaculture, for instance. Okay, so for instance, we have identified polychaetes, and particularly nerate polychaetes, as main candidates to explore this strategy. Why nerate polychaetes? Well, first, because they can culture intensively. Okay, so there is a good understanding on how to culture them massively. Okay, there is protocols established for species like, for instance, nerate virens, which is a polychaete that is produced, for instance, in the Netherlands, in Europe, I don't know, probably elsewhere as well, and it's used for bait, and also used for maturation diets for shrimp and broodstock, for instance. So there is a technology to grow these nerate polychaetes already in place. Polychaetes grow very well also on waste from different bioindustries, including aquaculture. Nerate polychaetes have also high adaptability to varying environmental conditions, and this enables us also to tweak conditions to switch on the activity of those omega-3 desaturases apart from other enzymes. And last but not least, polychaetes have a complete pathways, a complete set of enzymes enabling a complete pathways of long-chain purified biosynthesis. Okay, so apart from having omega-3 or omega-X desaturases, they also have front-end desaturases and elongases. So we are carrying out experiments using these two models that I showed you. And exploiting this strategy by which we aim to grow these animals under specific conditions to switch on the activity of the desaturases and elongases, endogenous desaturases and elongases, so that the biomass produced is enriched in omega-3, and this biomass eventually can be processed and be used as an ingredient for feed. Okay, so these are the two ongoing projects where we are developing this idea, one funded by the Spanish Ministry of Science and Innovation and Universities. It's called ImproMega. And the other one, the ImproMega project is applying this strategy in the nerate polychaetes, as I showed you earlier. And then a Norway funding project funded by the Research Council of Norway, led by SINTEF, a Norwegian company, that is exploiting this idea, but instead of using polychaetes, is using marine gametes to grow these gametes on waste streams from different bioindustries and produce an omega-3 rich biomass out of these gametes. Okay, so as concluding remarks, I don't finish my talk. Some take-home messages. Invertebrates possess desaturases and elongases involved in long-chain purified biosynthesis, so they have active networks of enzymes enabling them to produce these compounds. Compared to vertebrates, invertebrates have a higher diversity of desaturase and elongase families. As we saw the existence of different subtypes of elongases or front-end elongases. They also have omega-X desaturases, and they have these novel elongases that potentially can also contribute to the pathways we've been seeing. With regards to front-end desaturases, invertebrates have delta 5, delta 6, delta 8 desaturases, so a varied range of activities. Then they do have also purified elongases, including LOVL25 and LOVL4s, both involved in these pathways. And as I said at the last part of my talk, invertebrates, many of them possess these omega-X desaturases, enabling them to biosynthesize omega-3 long-chain purified de novo. Okay, so I'd like to thank you for your attention. I hope you learned something from this very biochemistry dense talk. Thank you so much.
Thank you so much, Dr. Mondrake, for your talk. So now it's the time for questions. I will turn off the recording now, so please feel free to ask your questions.